Welcome back to our YouTube channel Histrology. In this video, we will discuss this topic, the extracellular matrix, ECM. The ECM is an intricate and dynamic community of molecules outside of cells that afford structural, as well as biochemical, support to the adjacent cells in tissues. It acts as a vital player in tissue development, maintenance, and repair. Not only does it modify the mechanical characteristics of tissue, but it also influences activities occurring inside cells. It is made up of a number of proteins, glycoproteins, proteolycans, and polysaccharides that make up a very complicated scaffold that covers and interconnects cells within tissues and organs. This essay explores composition, structure, functions, and importance of the ECM, now centering around its critical role in being both healthy and diseased, specifically in relation to tissue regeneration and the process of cancer progression. Composition of the extracellular matrix. The ECM is predominantly comprised of three major classes of biomolecules. 1. Fibrous proteins. Collagens are the most abundant proteins in the ECM, giving tissues their tensile strength and structural integrity. There are more than 28 different types of collagen, with type 1 collagen being the most abundant. The collagen fibers are a triple helix structure that makes it strong and stable. They are structural components of the skin, bones, tendons, and cartilage. Elastin gives tissues elasticity, allowing them to snap back to their original shape after deformation. It is most important in tissues where they get stretched often, such as blood vessels, lungs, and skin. Elastin is an essential protein that gives elasticity to various tissues and enables them to spring back to their original shape in cases where they are stretched. It is more critical in tissues which are constantly elongated, such as those found in the walls of blood vessels, in the lungs, and in the skin. Fibronectin is a glycoprotein that enables cells to adhere to the ECM, and this clearly plays a crucial role in cell adhesion, migration, and healing. It also binds to integrins exposed on the cell surface and interacts with many other elements of the ECM, such as collagen and proteolycans. The principal proteins of the ECM basal lamina include laminins. These give fundamental structural support and regulate many aspects of cellular activity, including differentiation, movement, and survival. Laminins form interconnections with other elements of the ECM, like collagen 4, to generate a scaffold that provides structural support to cells. 2. Proteolycans and glycosaminoglycans. GAGs. Proteolycans are proteins carrying long chains of GAGs. These molecules are active in many different ways, including regulating molecular transport within the ECM and providing resistance to compressive forces. Notable examples of proteolycans are agrecan, which is plentiful in cartilage, and dicorin, which associates with collagen. Glycosaminoglycans are linear, unbranched polysaccharides with negative charges for the purpose of binding water. Such molecules, therefore, play a very critical role in making the extracellular matrix supportive to tensile forces and hydrating while modulating the activity of signaling molecules. Examples include hyaluronic acid, chondroitin sulfate, and heparin sulfate. Cell adhesion molecules are mediator activities between cells and the extracellular matrix. Integrin represents the primary cell surface receptor for binding to ECM proteins, such as collagen, fibronectin, and laminin. This allows signals originating from the ECM to bind into the cell cytoskeleton, thereby influencing shape, movement, and survival of the cell. The structure of the ECM is highly structured, with specific differences between different tissues being crucial to and necessary for particular functional purposes. The two major components of the ECM include 1. Basement membrane, basal lamina. This is a thin layer of ECM that is located at the bottom layer of epithelial tissues. It gives support to structure and restrains cellular activities because it acts as a point of contact between the epithelial cells and the connective tissue. The basement membrane has laminin, collagen 4, nidogen, and perlecan. 2. Interstitial matrix. This is part of the ECM that lies in the interspace between cells in connective tissues. Its density is less compared to the basement membrane, and it is rich in fibrous proteins such as collagen and elastin, together with proteolycans and GAGs. The ECM functions as an active, performing component rather than a static or passive framework. It continuously undergoes remodeling in response to cellular activity and mechanical stress as well as alterations in the environment. The class of enzymes referred to as matrix metalloproteinases enables ECM constituents to be degraded, thus opening pathways for tissue remodeling in growth, repair, or disease processes. Glycosaminoglycans, or GAGs, are long linear polysaccharides that are characterized by their power to attract and retain water due to their electronegative charge. Crucially, GAGs facilitate the resistance of ECM to compressive forces, hydration, and regulation of signaling molecule activity. The most famous GAGs are hyaluronic acid, chondroitin sulfate, and heparin sulfate. In general, CAMs are involved in mediating cell interactions with the ECM. Integrin is the principal receptor on cell surfaces that will bind to the ECM proteins of collagen, fibronectin, and laminin. The binding also initiates signal transduction in the bridging of the ECM to the cytoskeleton of the cell, which then determines the shape, movement, and survival of the cell. The architecture of the ECM is highly well organized and differs from tissue to tissue in a manner that appears to be adjusted to specific functional demands. There are two major constituents of the ECM, which are 1. Basement membrane, basal lamina. It is a thin sheet of ECM situated at the base of epithelial tissues. Being a barrier between the epithelial cells and connective tissue, it provides structural support along with controlling the behavior of cells. The basement membrane consists of laminin, collagen 4, nidogen, and perlecan. 2. Interstitial matrix. As its name describes, it lies between cells within connective tissues. It is much less dense than basement membrane and consists of rich fibrous proteins like collagen and elastin, together with proteolicans and GAGs. The ECM is not a passive scaffold but an active entity under continuous remodeling due to cellular activities, mechanical stress, and environmental changes. MMP's enzymes catalyze the degradation of ECM components, which aids in tissue remodeling during processes such as growth, repair, and disease progression. In the tissue repair after an injury, the ECM plays a vital role. Replacement of damaged ECM components. When tissue is damaged, local cells like fibroblasts replace the damaged ones by producing replacement ECM components. This is the very essence of good wound repair. However, improper regulation of the ECM during the repair process leads to fibrosis, whereby there is an accumulation of too much collagen resulting in scar tissue that denies the tissue its proper functionality. The heart of such research is artificial ECMs, or scaffolds that can be developed by scientists in the field of regenerative medicine to facilitate tissue regeneration. These scaffolds can actually mimic the ECM environment naturally, enhancing cell adhesion, proliferation, and differentiation to assist in regenerating damaged organs and tissues. The ECM is complex in its involvement with disease and varied. Changed levels of the ECM cause numerous pathologies ranging from cancer and fibrosis. 1. Cancer. The ECM completely reorganizes during the process of cancer development. The cancer cells secrete enzymes such as MMPs that degrade the ECM, thereby, the tumor acquires the possibility of invasion of adjacent tissue and further metastasis. Besides, the ECM affects the behavior of cancer cells. For example, the increased stiffness of the ECM due to higher degrees of collagen cross-linking has been associated with more aggressive characteristics of cancer. Moreover, changes in the ECM also have been demonstrated to affect the immune reaction to the tumor, sometimes by boosting and at other times by inhibiting tumor growth. Fibrosis, an overabundance of ECM, is the hallmark of fibrotic diseases, which endows tissues with stiffness and impairs their function. It commonly occurs within organs such as the lungs, in pulmonary fibrosis, liver, in cirrhosis, and heart, in cardiac fibrosis. This leads usually to fibrosis with both fibroblasts and myofibroblasts participating in the overproduction of ECM components, particularly collagen. Conclusion: the extracellular matrix is a vital structural element and essential for all tissues. It gives them their integrity and necessary biochemical signals acting upon cellular activities. Its elastic properties allow it to be flexible with the changing demands of tissues at different developmental stages, wound healing conditions, or states of disease. Understanding the nature, structure, and functions of the ECM is a very important aspect for developing novel therapeutic strategies aimed at targeting tissue repair and disease manipulation, including diseases such as cancer and fibrosis. Advances in the biology of ECM continuously enhance our ability to engineer its properties for the field of regenerative medicine and correct its pathological alterations in disease states.